bleeding disorders. We have seen, we discussed about hemostasis, bleeding disorders, and we have seen the involvement of certain disorders like AML, CML. What is AML and CML? Acute myeloid and chronic myeloid leukemia. Bucket's lymphoma is a AML or CML? Bucket's lymphoma. AML. See? That is CML. Okay. Lymphoid. Bucket's lymphoma caused by lymphocyte blastomas. Okay. Then we discussed about platelet functions, hemostasis, then coagulation mechanism. In coagulation mechanism, there are three pathways. What are those? We have intrinsic pathway, extrinsic pathway, common pathway, and conversion of fibrinogen to fibril. We discussed this. Intrinsic pathway will be detected by what test? APTT. APTT for intrinsic pathway, whereas extrinsic pathway by PT, and other tests for checking the common pathway and fibrinogen production. Then we discussed about all these things. I hope you already know the 13 clotting factors, bleeding time, clotting time, all these things. This is one part of the story, right? I just want to tell one more announcement. So what I felt in the hematology group is there are many non-MLTians actually. You know, when I say non-MLTians, there are some students who are left from this group, left from this course, but they are still in that group. For example, Jashanpreet is in first year now, but still Jashan is in this group. So I don't want such mismatches in the group. And also, for example, Sunil sir. Sunil sir is not associated with this institution, but he can see all these things, which is ideally not good. Okay. Say for example, I got a simple thing, no clashes. We conducted a VAC workshop, and I said I will give you a certificate. Parallelly, I got some requests from other groups stating those need certificates even though they are not registered in VAC. And I have good relations, so I need to do some under the table things which I don't like. So better what we can do, we are migrating hematology group to a new hematology group. That's why the current group was renamed as a blood studies. And we are not hurting anyone, everyone will be there, no one will left from blood studies, that is a backup group, but we will migrate to a new group. And that migration link I shared with your CRs and some students, and this is the new group link, which looks hematology, but this is a new group, keep in mind. It's an empty new group. Once take photo of this one and then you can share among yourself. Take a photo of this and share in your unofficial groups. Okay? Share in your unofficial group, not in official groups. Please. Don't share in any official group. Okay? Yeah, that's it. Thank you very much. Shubham will take care of this one. Shubham will share the details of this group, okay? Anyway, proceeding further. Enough. No need. No one should leave from blood studies. Okay? Everyone, internationals, everyone, no one will leave from actual group, okay? But the group will be, eventually it will be silenced. Maybe we will use for other activities, okay? I don't want to terminate that group because that group has an extensive repository of PDFs, notes, links, data, some messages, which I don't want to, leave that group. So that will be as a store, okay? It will be an archive and we will be in new group. Got it? For example, weekend works are in old group actually. So we need both the groups, but that will be silenced. Got my point? No, nothing. So it's for good cause. Keep in mind. That's it. That's it. Coming back to my problem. Okay, the things. Yes. So now I will show you some more depth things of these things. So this is a platelet. So platelet has many things. So platelet secreting some things like platelet secreting ADP, platelet secreting serotonin. We know what is the role of serotonin. What is the function of serotonin? Vasoconstriction during hemostasis. VWF, 1 milligram factor. What is the purpose of? Okay, here it is showing like this is dependent on platelet. Actually, VWS is secreted by endothelial cells of the blood vessels, right? So 1 milligram factor, then PAF, platelet activating factor, necessary for platelet aggregation, and thromboxane A2 also necessary for platelet aggregation. Then its not a hormone, it's not a chemical, but it's a cell membrane but has a property. So initially they thought it's a hormone or cytokine, and they said it has a platelet factor 3. They assumed this is a factor, might be a cytokine, and they thought PF3. But today we came to know PF3 is just the phospholipid membrane of the platelet. So what is the role of PF3 or phospholipid membrane? Listen, this phospholipid membrane has negative charge. Okay, now we need to mind this is blood vessel and whole blood is under movement. Happily, I will mark you absent. You can stay out. Yeah, that's it. You can go. Yes, it's become regular. That's the problem. Okay. Listen. When the electrons are in the flow in a wire, they will make an electric charge. That is called electromagnetic flux. Am I correct? Yes. Movement will give some charge. That is universal physics. Here also, a moving blood will cause a charge development, negative and positive charge. So inside the blood vessel there is negative and outside the blood vessel there is a positive charge. Okay. It is because of this charge our mobile screens are working that I discussed earlier. Okay. And there is a significance of this charge, hemostasis and platelets. One of the reasons why platelets are activating is get in contact with a positive charged surface. Get in contact with a positive charged surface can activate a platelet. Okay, because phospholipids are negative, platelet membrane, and this can trigger a platelet activation. PF3 can activate platelet activation by getting in contact with a negatively charged surface. Okay, so why this is important? I will let you know. Take a small glass beads, small glass pieces, inject glass pieces into the blood. Game over. Glass pieces has negative charge, platelets will get activated and platelets will be clotted. The blood will be clotted. Okay? See, just a foreign subject can kill the patient. Like a glass beads in the circulation. Silica gel beads. Okay? That's why, fine, that I will discuss later. Most of the venom and poisons, they will create these charges. They will disrupt the platelet activities, or simply they will clot the blood. The idea of toxins or poisons, when the snake biting, the snake has only one agenda, to clot the host blood. If I clot the blood, that's it, it will die. And it's not a dangerous thing. Okay, clotted blood, it can eat it, because it need to eat that host later on, right? So it cannot inject a real toxins. It is indirect ways of killing them. Anyway, that is it. Other things I need to discuss was the receptors. There are two receptors. What are the two receptors we have? GP1B and 2B3A. Where is 2B3A in this? 2B3A missing. 2B3A. Yeah. So 1B is responsible for attachment of platelet to the 1 milligram factor, whereas 2B3A is responsible for aggregation of the platelets. So receptors you understood. Then I just want to show you hemostasis again in a different way. So this is how they are interacting each other. So hemostasis is not a simple task. It has many things. See here, there are multiple cytokines and multiple genes are there doing the hemostasis. But we are trying to study in a simpler way for better understanding. But this is how the hemostasis works. Okay. Yeah, we are not going in depth of understanding hemostasis. This is not we are not physicians. We are just doing the diagnostic parts. So we need not to worry about complicated things in the hemostasis. In a nutshell, what I need to introduce in the previous lectures is the involvement of PF3 in the coagulation mechanism and in hemostasis, both at the external to the platelet cell. During clotting mechanism, PF3 plays a vital role, and I will discuss PF3 role in the later lectures. Okay, so this is one important addition I want to add, and a quick overview of hemostasis: blood vessel, collagen fiber, 1 milligram factor, then platelet attachment GP1B, Then platelet will release thromboxane A2, serotonin, ADP, then 2B, 3A. Okay. Now, artificial 1 milligram factor. If I need to trigger a platelet artificially, listen, when I say artificially, like this: in vivo, in vitro. In vivo inside the body, and in vivo outside the body. In vivo 1 milligram factor. In vitro, we have a stimulator, a chemical called desmopressin. This I am adding today. Desmopressin, DDAVP. This is an injection that is used to stimulate the platelets activity in the patients. Okay, this is one in vitro chemical. And other one, thromboxane A2 or ADP or serotonin. These are in vivo chemicals. We know their purpose. Their purpose is for aggregation, right? And in vitro equivalent of this is ristocitin. So I have included new things here. Then high molecular weight kininogen, that is responsible in the intrinsic pathway where there is a high molecular weight kininogen. The similar chemical in vitro of high molecular weight kininogen will be cephalins, phospholipids, and there is one more chemical I forgot. There is one more that I will see later. Okay, so keep in mind these chemicals, and I hope you are understanding where these are connecting in the hemostasis. Clear, right? Everything clear, right? So far, yeah. Next one. So this knowledge you understood. The other problems with bleeding. Let us assume what lab test you will perform when you suspected bleeding or clotting disorders. First you are performing bleeding time, clotting time, then you are going with CBC. First you might perform CBC, then you went for PT, CT, and what else you will do? Yeah, coagulation profile you are doing. PT, APTT, TGT and TT you might do. Then what else? Platelet function test. Okay, platelet function test. Today's lecture is about platelet function test actually. Okay, so in platelet function test, what test you will perform? Adhesion tests, aggregation tests, secretion tests. Okay, activation or secretion tests, right? Then what else you can do? Maybe here only you have other things. What is the role of flow cytometry? What is the role of flow cytometry? Do you remember what is flow cytometry? Yeah, what is the role? So it will study the cells in the movement, and it can also study the cell receptors by using a laser light. The forward scatter will check the size of the cell, whereas the side scatter light will check the complexity of the cell or the receptors of the cell. Do you remember? So we have two receptors, we already know, 1B and 2B, 3A. 1B deficiency is called as Gallman's thrombocythemia, whereas 2B, 3A deficiency is called as Bernard-Solier syndrome. I said in vice versa, sorry. Bernard-Solier syndrome, Gallman's thrombocythemia. Clear, right? Now, these receptors can also be verified by electrophoresis method. These receptors can be checked by electrophoresis. Electrophoresis will separate the protein molecules, if you remember. Have you studied separation techniques in the biochemistry? Chromatography techniques, what is that? Thin layer chromatography techniques, HPLC. Yeah, so then patient, one more, what is patient? Other one? SAGE. What is PAGE? Polyacrylamide gel electrophoresis. SDA. I forgot. Southern blotting. Okay, anyway, there are multiple electrophoresis methods. We are using electrophoresis to separate the proteins and we will cross verify there. Okay, that is the role of electrophoresis. Then, now into this equation, we need to bring the fibrinolytic mechanism also. Fibrinolytic mechanism. Okay, hemostasis won't end with the arrest of bleeding. Hemostasis also continues to removal of the clots from the circulation. Do you remember? The clots will be removed. So in fibrinolytic mechanism, there is a scissor molecule. What is that? Plasmin. Very good. The plasmin, and there is one more D-dimers. Okay? Degraded fragmented D-dimer molecules. They are also scissor molecules. Now listen. If plasmin and D-dimer are more, obviously again we will get a more bleeding time. Okay? Their levels can indirectly cause excessive bleeding time when there is an excessive fibrinolytic mechanism. If patient has all normal values, when we performed all these investigations and all these came normal, then we need to check antagonistic tests. Antagonistics means they will act as an agonist to bleeding. They will promote bleeding actually. They won't stop bleeding, they are promoting bleeding. So we will do tests for level to detect these levels. D-dimer levels and FDP. Fibrin, degraded products we will check, and plasmin we will check. Right? Parallelly we can also check PF3 levels. Okay? Yeah. Now, is there any cheap and alternative test to know the overall efficiency of hemostasis in the body? That is the primary problem. When we are going for individual investigations, lab test will cost around 25,000, 25 to 30,000, and we cannot do 30,000 tests for each and every patient. Keep in mind, cheap and no, that is the first priority. It should be cheap, it should be efficient, it should be simple. In that case, is there any alternative test to check the overall efficiency? Sometimes what happens is, sometimes every values will be normal. When we are seeing individual values, all values will look like a normal values. But patient exhibiting some bleeding condition. Patient showing some bleeding problems, but his things looking normal. What might be the reason? The reason is the overall system rigidity. What is the meaning of overall system rigidity? Imagine for a car we need engine, everything we need, right, to make a functional car. Now let us imagine car has a V12 engine, Lamborghini engine. So everything is good, but the bolts are not tight. Maybe they are not integrated properly. Maybe they are not communicating each other properly. That can cause a collapsed car. That might cause some defects in the car performance. Okay? How to check the overall efficiency of hemostasis? That is the primary problem to the scientists. When individual values are same, they are fine. But when they are checking together, there are some issues. How to check that test? And they came up with a simple solution. Let us perform these activities on a slide outside the body. Again, that might be a costly effort. How can we do hemostasis studies outside the body? Complete system should be involved in it. They came with a very simple solution. They took a test tube. Okay. They took a separated tube. So coagulation factors are nullified. Okay. They took platelet poor plasma. They added platelet poor plasma. To this, they added platelet rich plasma. Now they purposely added platelet rich plasma with an antagonist, maybe with aspirin. With the help of aspirin, they blocked the platelets. Aspirin will block the thrombexin A2 receptor. So aspirin blocked the platelet activity. Now this platelets are added to the plasma to the same patient. Now what they did is they added a stimulator. It can be a ADP, epinephrine, or serotonin, thrombexin A2, or ristocetin. I said right? Ristocetin are added, or desmopressin, DDA, VP. Upon adding any one of the stimulator, what happens? platelets slowly aggregate, okay, they are activated, they are aggregated. Now aggregated platelets, see in this is a CBC, whole blood. Okay, we added plasma, then we added whole blood along with platelet rich plasma. Okay, now theoretically what should happen is platelets should be activated. They must convert fibrinogen to fibrin. They must, trap the red blood cells. They must trap the red blood cells. That is a phenomenon of clotting, right? Now there is an in vitro hemostasis observed. In vitro platelet plug formation has been observed. Okay, what I will do is I will take a iron loop. I kept a spring at the end. Then I will lift the iron loop. I will see how much platelets are aggregated. Okay, so now HCT. What is HCT means? Hematocrit. What is the role of hematocrit? That is also PCV. That is the volume of red blood cells to the plasma ratio. Right? Ratio of red blood cells to plasma. When there is a good response, a normal platelet response must show a decreased HCT. You agree with me? A good platelet response must reduce the HCT values maybe after 4 hours. Because all the blood cells has been clotted, they trap, so plasma to red cells ratio will be reduced. A good platelet performance must reduce a HCT. And directly proportional I will write. Thrombocytopenia. Reduced platelets are directly proportional to HCT. Decreased platelets, increased HCT. Okay. Actually, this is also inversely proportional. Okay. So decreased platelets, increased HCT, and normal platelets must decrease the HCT. This is called as clot retraction test. When we suspect an abnormal clot retraction, this is called clot retraction. Keep in mind. Okay. After clotting, the cells has been, the elasticity, how can I say? They are clumped together. Okay. So that is called clot retraction. Now, this test will give other important things. It will also check the elasticity nature of the fibrin clot, how much tight it is or how much fragile it is. I can check the elasticity property of the clot. That is one interesting insight. Elasticity I can check, plus other problem. If there is no proper elasticity, but there is a good rigidity, it was very rigid. These two properties are very important. Elasticity and rigidity of the formed clot. Okay. Then last and final, the flow of the blood. This is called as shear pressure. What is the meaning of shear pressure? I will discuss. Shear stress. They are saying shear stress. What is the meaning of shear stress? Shear stress means this is the pressure activated on fluids that are in movement. In the blood vessel, the fluids are moving, right? And that moving fluid will exert a pressure. That pressure is called a shear pressure. Now imagine when you have a blood cut, platelets are working against these pressures. Platelets are tolerating these pressures. Platelets are tolerating these shear pressures and they are blocking the bleeding. That need to be considered. What is the shear pressure tolerance of a platelet clot? Platelet primary plug shear pressure tolerance. How to check these parameters? We need to measure these parameters also. These parameters cannot measure by a clot retraction test. Now I will give one simple hierarchy. I know bleeding time is best to know platelet activity, but bleeding time is not so good compared to clot retraction test. But clot retraction test is not so good to identify elasticity, rigidity and shear pressure factors. Got my point? This clot retraction cannot check my elasticity nature and shear pressure, and it cannot check my rigidity of the platelets. So we need a new test. We need some new equipment to measure these properties of the platelets. Previously we thought just receptors, 1 milligram factor, those are sufficient. But when we are clubbing everything together, still there are some problems. We need to see the overall integrity of the hemostasis phenomenon. And that is where today's story will start. This is all about introduction. And this is where the story starts about platelet function tests. Yeah. Now, what is clot retraction test? Okay, listen. Clot retraction test will measure the overall performance of platelets, starting from platelet activation to platelet clot formation. Okay, that's it. Yes. Yeah. Now write down. I have already shared clot retraction notes with you. So, ideally, clot retraction normal values. Write down. Clot retraction test normal values. I hope you understood what is clot retraction. We are adding plasma, platelets, platelets, some stimulator. Then we are clotting them and we are checking after some time by lifting the spring to see the clotted thing. Okay. I have an image here. I will show you. 1 second. Yeah, this is clot retraction. See, students, a normal blood, then after clotting, red blood cells are shrunken. Why cells are clumped and shrink? Because that is what clot is all about, right? The hit CT will be reduced. The plasma to cells ratio reduced in clot retraction test. Now, there are multiple approaches of doing clot retraction. And this is not a precise test for any condition. This is just a non-specific test for understanding the hemostasis phenomenon. This is not a gold standard test. Keep in mind. Just you got an abnormal clot retraction doesn't mean patient has hemophilia or any other condition. This is just like a ESR. Got my point? Yeah, this is clot retraction. See, there are various stages of clot retraction. Heavy retraction, small retraction, negligible and nothing. So there are some stages of clot retraction. So ideally, write down a normal value. Approximately 30% of total volume should be clot. Approximately 30% of total volume of blood must be clotted in a normal condition. Okay, 30% of blood must be clotted in normal condition. Now, in case of thrombocytopenia, write down in clot retraction test, in case of thrombocytopenia, that means reduced platelet count, what might be the expected results? Guess, students. Platelets are reduced. What will be the clot retraction? What will be the clot retraction? In case of decreased platelet count, there will be a less clot retraction. Got my point? Very good. Yeah. In case of in case of thrombocytopenia, large clot with less retraction. Large clot with less retraction. The blood won't be retracted because retraction is caused by platelets, but blood can clot because clotting happens by clotting factors. Got my point? So there will be clot of blood will be clotted, but there won't be any retraction. Why there is no retraction? Because less platelets. Got my point? Second one, what will be the condition in case of a fibrinogen deficiency? Decreased fibrinogen. What will be the clot retraction? Tell me. Fibrinogen reduced, but normal platelets. Now tell me. Platelets are normal, but fibrinogen reduced. So, less clot, but heavy retraction. There will be huge retraction of blood, but the clot, the consistency will be less. It is not clotted. It will be fluid state, but there will be good retraction. Got my point? See, just a simple clot retraction giving deep insights. Okay? Then, enhanced fibrinolysis. Enhanced fibrinolysis means there is a huge fibrinolysis in the body. Fibrinogen is there, but fibrinolysis, maybe D dimers are more. D dimers, FTPs and plasmins are more. In this case, what happens? Platelets are normal, they contracted and clot happened, that means there is a thick clot, but also FTPs are smashing the clots. So the quantity of the clot will be less, like this. Here, the quantity of the clot will be reduced in case of excessive fibrinolysis. When there is excessive fibrinolysis in the body, that means D dimer or FTP is present, plasmins D dimers, platelets retracted properly, clotted properly, but D dimers destroying them immediately. Okay, maybe the standard time might be 4 hours for this test. After 4 hours, when there is a less clot, that indicates there is a heavy D dimer activities, fibrinolytic activities in the blood. Okay, now listen, this is just an analog technique, that means a manual technique. Still I cannot study the rigidity status of the clot, the elasticity status of the clot, and the overall efficiency of the platelets. I cannot study the minute measurements of platelets. Even the clot retraction giving me some deep insights. Got my point? I need a scientifically accurate results. That is where again the next generation test came. Now I will discuss that. Write down. So platelet function tests. Types of platelet function tests. Write down. Types of platelet function tests. Types of platelet function tests. 1. Write down in number sequence. Bleeding time, platelet aggregation test, platelet adhesion test, bleeding time, platelet adhesion test, platelet aggregation tests, then platelet secretion test, platelet secretion test, then flow cytometric analysis, flow cytometric tests, then PFA 100 test, PFA 100 test. I will tell what is PFA 100 test. Okay, PFA 100 test, platelet function assay test. That is a equipment. We have CBC analyzer, right? What is the analyzer name? H560, EPA H560. So we have a special reader called PFA 100. Then one more, write down. Cone and plate analyzer, CPA. Cone and plate analyzer. It is also called as impact test. Impact test. These are the actual tests using in labs now. Whatever we discussed are old techniques. Okay. CPA. Cone and plate analyzer. Cone and plate analyzer test or impact test. One more test, write down. Cone and plate analyzer test. Ultra rapid platelet function assay. Ultra rapid platelet function assay test. Ultra rapid platelet function assay test. Ultra rapid platelet function assay test. And okay, I didn't wrote the full form of this one. I will check in the PPT. Okay, I forgot. This test is called as HAS test. This is the best technique, students. HAS test. All these are auto analyzers, by the way. All are auto analyzers. Just you need to be sample and it will do the report automatically. HAS test. HAS test. HAS test. Okay. Maybe in tomorrow's lecture, HAS. Okay. Hemolytic something. Chemo something test. I will let you know later. Platelet aggregation test, ultra rapid platelet function test, CPA, cone and plate test, flow cytometry, platelet aggregation, platelet secretion test. In case of platelet secretion, we are checking the thromboxane A2 and arachidonic acid synthesis test. Arachidonic acid synthesis test actually. Okay. What is the time exactly, tell me, students? 52. I will take 3 minutes more, then I will finish it. Yesterday in the group, I have shared platelet function test review paper, a research paper. From that research paper, I have collected the notes. Okay. If I am asking platelet function tests in the exam, you must write the notes from that paper. Okay. I will share that paper again. Anyway, let us do a quick summary. Yes, students. So this is of one technique. Actually, during Ranjay sir, he said he will provide a grigometer. Okay, but that is very costly. We didn't purchase it, but he was very interested. But I am not interested because you might damage it. That's very costly, around 4 to 5 lakhs. Just for clotting studies, we need 4 to 5 lakhs. When we have BTCP, that is sufficient to you. Anyway, I'm sorry, I'm not demotivating you anyway. This is how it will work. Blood will be clotted, or platelets will be aggregated. That will change the turbidity nature of the plasma. Via light, we will check the turbidity. Turbidity is directly proportional to aggregation activity of the platelets upon adding some apparatus. Okay. Now, I will show you one test. The first test is this one. What is the first test? PFA 100 test. In PFA 100, what we are doing is, in PFA 100, for all platelet studies, we need to take the platelet-rich plasma. Keep in mind, platelet-rich plasma will be taken for platelet studies, whereas coagulation studies we will take platelet-poor plasma. Coagulation studies we are taking platelet-poor plasma, whereas for platelet function test we will take platelet-rich plasma. We will take purposefully platelets, abundant platelet-rich plasma. Platelet-poor plasma for coagulation studies. Okay? Yeah. Now, first I will get my PRP, platelet-rich plasma. That's it. Simply I will centrifuge the bag. So upon centrifuging, I will get a sediment that will have platelets. I will take it. Okay? Now, to this platelet-rich plasma, I will keep an antagonist to block the platelet activity, maybe aspirin. Okay? Aspirin will block the platelet activity. Then I will add the activator. Activator can be a ristocitin or DAVP. Then I will do the test. The PFA 100 test is very good for detection of hemophilia. 1 milligram factor deficiency. Write down. PFA 100 test is a gold standard test for detection of. PFA 100 is a gold standard automatic, semi-automatic method. PFA 100 is a gold standard semi-automatic method for detection of 1 milligram factor deficiency. 1 milligram factor deficiency. It works on the principle of development of turbidity upon aggregation of platelets by optical measurement. Okay, it works on the development of turbidity by optical measurements by platelet aggregation. Okay, yeah, PFA 100. Other things you write down. Results will be obtained within 6 minutes. For PFA 100, I am discussing about PFA 100. Results will be obtained within 6 minutes of the test. Results will be obtained within 6 minutes of the test. Okay. Sample size, blood sample volume. Sample volume is 0.12 microliters, 0.12 ml. 0.1 ml is sufficient to get the results. 0.1 ml. Okay. Now, other advantages, it is rapid. It is a rapid, easy to perform test. It is a rapid, easy to perform test. Rapid and easy to perform test. Maybe tomorrow I will continue, okay? This Saturday you have a classes to know, right? And also next week you have exams. Next week from when? Next Wednesday or Monday onwards? Monday. 20. Wednesday onwards, right? Next to Wednesday onwards, not Monday onwards. Keep in mind. It was changed now. Okay, thank you very much. Until now, listen, class, listen. You need to go through the notes, okay, after you are going to home to understand these topics. You need to do work from your end to understand. And also, please write weekend works on time. Weekend works notes is very simple notes actually. You will understand easily these things. Thank you very much. Yeah.